Hey everyone, this is Elad from S2 Lab Diagnostics, and today I would like to discuss what is an analysis pipeline and how to develop one for your high complexity cytometry data, whether it's CyTOF, Cytocorora, a 14 color for Tesla experiment, or anything else. So what's an analysis pipeline? You have your instrument and you use it to get some single cell data, but that single cell data doesn't have biological meaning. It's just a bunch of biaxial plots. And what you would want is to get some meaningful biological insights out of it. The analysis pipeline are these question marks. This is the connection between the raw data and something that has context, that has significance, that has value from a biology or immunology perspective. And an analysis pipeline is not something new. In fact, it existed since statistics existed. And you can talk about these steps in the 40s and the 50s. It has garnered some more attention due to data science. However, once again, it is not new to data science either. So the steps I'm showing you here, they are not an invention of computational biology. They have a pretty long pedigree. And a pipeline says that you start from your raw data and you clean it, you convert it into meaningful features, and then you run some sort of statistical analysis. On top of all of this, you have a layer of different visualizations that help you make sense out of each one of the steps. And in the context of high complexity cytometry, building a pipeline is a bit like answering a multiple choice questionnaire, where for each one of these steps, you can choose one or more methods or protocols or algorithms to help you fulfill them. For example, for cleaning the data, you can use debarcoding, you can use an event length versus DNA or Gaussian parameters for the CyTOF, you can use forward scatter versus side scatter for flow cytometry. Then for feature generation, you can either use traditional gating, you can use different clustering algorithms, or you can use the hyperspheres algorithm, and so on. And finally, for the statistical test, you can use t-test, logistic regression, GLMNet, and assorted R packages, ANOVA, and so on and so forth. Finally, for visualization, one of the options is using dimensionality reduction, such as UMAP or TSNE, or you can use traditional figures, biaxial plot, heat maps, box plots, and so on and so forth. So to build your pipeline, you can go through each one of these boxes and choose the relevant lines or the relevant algorithms. Of course, reality is a bit more complicated than just making these choices. You also need to implement them and make sure they're connected. But from a high level view, this is what you need to make sense out of your data. And I'm going to give one example pipeline from this paper from Michlmayr et al., which was published in Molecular Systems Biology last year. And in this study, 42 pediatric patients who were sick with chikungunya showed up at the clinic in Nicaragua, and blood samples were taken from them during two stages of the infection, during the acute and convalescent stages. And this is an immune monitoring study. The goal is to compare the structure of the immune system between these two time points. The study included different assays, and I'm going to focus on the high complexity cytometry one down here, where they've taken the PBMCs and used I think a 33 panel mass cytometry experiment to acquire them. So they got the data, and then what have they done with it? So first of all, first step is cleaning, and these are pretty much copy pasted from the paper's method section. They remove normalization beads, they identify cells based on DNA, they remove doublets based on DNA and event length, and they remove dead events based on RH103. Likewise, for flow cytometry, there is no normalization beads, but there is compensation, and cell events and doublets will be identified on forward scatter and side scatter, and dead, alive, with one of the dyes for dead or alive. Then it's about feature generation, taking the single cell data and making it into something that has a biological name that makes immunological sense. And what they've done here is they use a combination of methods. So first of all, they use this method called node labeling that starts from supervised clustering and then fits a logistic regression model to the supervised clusters, basically traditional gating. So they use that to identify the major components in the immune system, which are labeled here. So MDCs, PDCs, CD4 plus T cells, and so on. Then they took each one of these compartments and clustered them using unsupervised clustering. And for that, they used the method called hybrid Louvain, which is the underlying algorithm for phenograph. And finally, in order to connect the different samples to each other, they used meta-clustering across these different samples. And the end result is presented here using a Visney map. So this also connects us to the visualization within dimensionality reduction. Each one of these dots is one of the I believe one of the clusters that came from one of the samples. The labels are the traditionally gated cell subsets that were then trained using logistic regression, and the numbers are the clusters that were found using unsupervised clustering. So a bit complex, but all the details are here. And finally, for statistical analysis, they used a variant of t-test. They just they started from t-tests, and then they used a, a multiple testing correction to get an FDR score. And what you're seeing here are each one of the clusters that they found and the log 10 of the fold change between the two time points. So we started with the single cell data. We started with this mess of dots and biaxial plots and so on, and we ended up with this figure. And this figure is all about biological meaning. It's all about immunology. And it tells us, for example, that if we look at the far right, there is an increase of classical and intermediate monocytes within the acute time point. And on the other side of the spectrum, you can see different MDCs, you can see another population of classical monocytes, and you can see quite a lot of lymphocytes. So there seems to be a transition here where the acute time point has a lot of innate immune cells, while the convalescent time point has a lot of adaptive immune cells. So we started from single cell data, and we ended up with something that has a lot of context and meaning. And here is the answers that the authors of this paper have chosen for their multiple choice questionnaire. For cleaning, they use these traditional gates. Then for feature generation, they use a combination of traditional gating and phenograph and metaclustering. For statistical analysis, they use fold change and t-test. And then for visualization, they used traditional figures and dimensionality reduction using t-SNE. So again, this is just one way to build your analysis pipeline, and there are many such papers right now, several such manuscripts available that either talk just about the pipeline, such as Nowicka et al. At F1000, or papers with more biological meaning that include these elements in their method section. So I hope this makes sense. Feel free to reach out if you have any questions. If you are stuck on building your own analysis pipeline, then Astrolabe has an analysis pipeline which is ready for you. You can just give us your FCS data, and we're going to take it through all the steps that I just outlined. So if you need help with your analysis, please reach out at elad@astrolabediagnostics.com.